The overall goal of the following procedure is to isolate and quantify peptides in a complex mixture that act as surrogates for their respective protein. First, large proteins within the mixture are digested to component peptides using an enzyme such as trypsin. Then, the targeted peptide analytes and a spiked stable isotope-labeled internal standard are enriched using anti-peptide antibodies immobilized on protein G-coated magnetic particles. Following isolation, the targeted peptides are eluted from the magnetic particles and analyzed by mass spectrometry for quantitation relative to the internal standard.
The main advantages of this technique over existing methods, like traditional immunoassays, is that it is faster and more cost-effective in building large numbers of assays, it has a high success rate, and it is readily multiplexed. Demonstrating the procedure will be Leigh Zhao, a technician in the laboratory.
Thaw a 10 microliter plasma aliquot on wet ice. Transfer the sample to a 1000 microliter deep well plate and cover with pierceable film. To denature the proteins, add 20 microliters of fresh 9 molar urea 30 millimolar DTT to each sample and incubate for 30 minutes at 37 degrees Celsius. Add 3 microliters of fresh 500 millimolar iodoacetamide to each sample and incubate for 30 minutes in the dark at room temperature. Now, dilute the urea to 0.6 molar with 100 millimolar tris pH 8 and add 10 microliters of 1 microgram per microliter trypsin stock solution for a 1:50 enzyme to substrate ratio.